All right. If everyone wants to take their seats, we can look to get started. So welcome to our weekly orthopedic surgery grand rounds. This week, we have a new faculty member, Dr. Michael Chow, who will be here to present. Just also want to say congratulations again to our chief residents. Bones Ball was great on Friday night, and I think everybody had fun celebrating. The presentations at Inman Abbott were awesome. It's always fun to see everything that's happening in the department. So congrats to everybody who presented on another great meeting. And for introduction today, I'll turn it over to Dr. Pandya.
Thanks, everyone. I have the honor today of introducing my new ped sports colleague, Dr. Michael Chow. Michael's one of our 4 new pediatric orthopedic faculty that we hired this year. I know you heard from Dr. Hill. We've got Dr. Gephardt in the room and Dr. Ramirez, who's on maternity leave. One of the criteria I had for hiring someone was that me and Michael combined are under 200 pounds. So we needed someone my size. But very, very excited to have Michael join our faculty. Michael is a pediatric orthopedic surgeon who specializes in pediatric sports medicine. One of the things I remember Dr. Ma telling me years ago is you always want to hire people who are better than you. And if you hear Michael's background, he truly is amazing in terms of his educational background and his research interests. So Michael had a bachelor's and master's from Northwestern. Then he got his doctorate in medical sciences on cartilage biology in Sweden. Then he got his medical degree from Penn State. Then he did his residency at University of Minnesota. Then he did 2 fellowships. He did his peds ortho fellowship down at Scottish Rite in Dallas, and then did his sports fellowships at Curl and Job. So clearly tons of training, tons of experience. One of the things I've been most impressed about Michael since he started is just how thorough he is, how much he thinks about his cases, and just kind of the degree of scientific thought he's brought to our pediatric orthopedic group and pediatric sports practice. So Michael will be talking about the love-hate relationship with chondrocytes, which I'm really excited to hear about. So without any further delay, here's Dr. Chow.
Well, good morning, everyone, and thanks, Nora, for that kind introduction. So as a new attending, I, having just started practice last September, I figure I wanted to spend this time sharing with you my story of how I got here today. So I hope this will resonate with our trainees as well as those who are interested in musculoskeletal basic science research. My story essentially began as a love-hate relationship with the chondrocyte, which, as we know, is the cell that synthesizes cartilage. Because this was the focus of my research ever since I started researching over 20 years ago. It ultimately, I think, represents, for me, the challenges one faces or one can face in pursuit of becoming a surgeon-scientist-educator, which some believe is an endangered entity in today's environment. I have no conflicts of interest relevant to this presentation. The objective of my talk are to, 1, introduce myself as a new attending and share my personal journey to orthopedic surgery. 2, discuss challenges for aspiring surgeon-scientist-educators beginning in orthopedic practice. And 3, share my research interest related to the subspecialties of pediatric orthopedics and sports medicine. Nora, I've already kind of gave some of my background, but here's a little bit of overlap. Starting with a bird's eye view, my journey began on the West Coast. I ventured to the Midwest and East Coast for undergrad and medical school, respectively, and then to Europe for a doctoral degree. The reason for my decision to go abroad was largely due to my trust and friendship with my mentor. His name is Dr. Ulla Nilsson, and my passion for the research that I was conducting at the time. It also helped that there was a common thread between cartilage and orthopedic surgery, which was a specialty I had already set my mind on at the time. Furthermore, as it turns out, cartilage comprises both growth plates in children and joint surfaces in athletes. Therefore, after residency back in the Midwest, I completed a pediatric orthopedic fellowship in the South, followed by a sports medicine fellowship on the West Coast before joining the faculty here at UCSF. The chondrocyte. In most subspecialties of orthopedics, with the exception of oncology, we rarely look at individual cells. orthopedic surgery is predominantly a macroscopic practice with the goals of restoring or purposefully altering gross anatomy. For me, before I became an orthopedic surgeon, I was a basic scientist studying the chondrocyte, the single cell type found in cartilage. I started at the National Institutes of Child Health and Human Development in the lab of Dr. Jeffrey Barron. And to be honest, I'd always wanted to be taller because I love playing basketball. So the thought of studying growth plate cartilage was very intriguing to me at the time. How does the body know how much to grow and when to stop growing? How does an elephant grow to be so much bigger than a mouse? What causes the hundreds of different types of skeletal dysplasias we know in orthopedics? These are all questions that remain elusive and continue to be fascinating in my mind. My initial research involved studying the cellular and molecular mechanisms regulating longitudinal bone growth at the level of the growth plate. So we know this. Long bones grow by a process called endochondral ossification, in which cartilage is first formed and then replaced by bone. Initially, mesenchyme condenses and differentiates into cartilage templates composed of only one cell type, the chondrocyte. Chondrocytes at the center stop dividing and increase in size, and these so-called hypertrophic chondrocytes direct calcification, track blood vessels, bone cells, and ultimately undergo apoptotic cell death to give rise to primary ossification centers. At the time, around the time of birth, a similar event occurs in the middle of the epiphyseal growth cartilage, giving rise to secondary ossification centers. An expansion of these bony structures eventually compartmentalizes growth plate cartilage centrally and articular cartilage peripherally. Linear bone growth continues through chondrocyte proliferation, differentiation until the end of puberty when growth plates fuse. And finally, in mature long bones, cartilage persists only at articular surfaces or contributes to long bone maintenance by reducing friction and distributing loads. We also know that growth plate cartilage is structurally subdivided into three chondrocyte layers: resting, proliferative, and hypertrophic zones. A stepwise differentiation program occurs whereby stem-like resting cells give rise to highly replicating proliferative cells, which in turn increase in size to become hypertrophic chondrocytes that ultimately undergo apoptotic cell death and are replaced by bone. Articular cartilage can similarly be subdivided into superficial, intermediate, and deep zones. However, the differentiation program is less clear. In both growth plate and articular cartilage, chondrocyte proliferation and differentiation are regulated by local paracrine signals, which are in turn likely modulated by endocrine signals. So we have all heard of and maybe even use bone morphogenetic protein in our practices. BMP signaling accelerates longitudinal bone growth by promoting chondrocyte proliferation and hypertrophy. And the BMP ligands, the receptors, antagonists, at that time, when I was studying this, had been implicated to be produced by an inside source within the growth plate. This was my first ever biomedical research project. This is a proximal tibial epiphysis in 7-day-old rats that had been microdissected into chondrocyte layers under a dissection microscope using needles and razor blades. And then mRNA was extracted and was transcribed to cDNA in order to run microarray and real-time PCR to study BMP gene expression across the growth plate. In this project, it was concluded that BMP signaling gradients play a key role in the spatial regulation of chondrocyte differentiation, as depicted in this diagram. As I recall at the time, this project, although very interesting, was also very painstaking and time-intensive. And as a practicing orthopedic surgeon, I can't hardly foresee doing something like this today. There are a myriad of other molecular signaling pathways in the growth plate. In brief, this is the Indian hedgehog PTHRP feedback loop that determines the boundary at which proliferative chondrocytes stop dividing and begin to hypertrophy, and thereby controlling the length of the proliferative chondrocyte columns. At the time, while this had been known to occur in embryonic growth cartilage after birth, when the epiphysis undergoes major structural changes with the formation of secondary ossification centers, this mechanism, the structure and organization of it was not as well known. So this project, therefore, sought to better define the organization of the Indian hedgehog PTHRP system in the postnatal growth plate. First, microarray and real-time PCR analyses were used similar to before with growth plate microdissection into the various zones. And second, chondrocytes showing active Indian hedgehog signaling were identified using a transgenic mouse model in which the coding sequences of GLE1, which is that figure in green, acting downstream of Indian hedgehog, were replaced by a gene called LAGC. In turn, LAGC codes a bacterial enzyme called beta-galactosidase that reacts with a substrate called XGAL, which stains the cytoplasm of cells blue. Hence, all the blue cells you see here demonstrate active Indian hedgehog signaling. These findings collectively supported the hypothesis that the embryonic Indian hedgehog PTHRP feedback loop is maintained in postnatal growth plate. To me, at the time and even now, it's one of the really cool things about basic science, which is that there are so many different creative strategies, techniques you can use to test hypotheses and answer questions. This also means that there is not only just one way of doing something, but multiple different solutions. So, in between my second and third years of medical school, I made the decision to take a leave of absence to continue my research and pursue a doctoral degree. I followed my mentor to his home institution in Stockholm, Sweden, called Karolinska Institute. And at the time, it was somewhat bittersweet because I had left behind my med school classmates. But I saw it as a once-in-a-lifetime opportunity, and it made sense at the time for me to follow a common theme of cartilage research and to pursue my passion. So while abroad, I sought to address the following question. So assuming articular and growth plate cartilage are discrete tissues, but arise from a common cartilaginous condensation, hence similar mesenchymal stem cells. And given that they have comparable spatial architectures consisting of distinct layers of chondrocytes, what molecular signaling pathways, if any, regulate their divergence in the same cell type? This study used microarray and bioinformatics to compare gene expression in individual cartilage layers, 10-day-old rat tibias at the age when the secondary ossification center are just becoming apparent. There are various analyses, including bioinformatics, which are beyond the scope of this talk. It was found that many genes that were upregulated in the intermediate and deep zone, the green shown on top, overlapped with those in the resting zone of the growth plate, the green outline in the bottom. Furthermore, superficial zone articular cartilage showed gene expression profiles similar to both the proliferative and the hypertrophic zone of growth plate cartilage. Given this finding, we then hypothesized that if subject to the ideal microenvironment and orientation, that growth plate cartilage may be able to differentiate into articular cartilage. So that was the hypothesis. In the final project of my doctoral studies, we took osteochondral allografts consisting of articular cartilage, epiphyseal bone, and growth plate cartilage from distal femoral epiphyses of rats. These are essentially oat cylindrical plugs, rats expressing green fluorescent protein, and transplanted them to matching sites in wild-type littermates, thereby allowing tracing of the transplanted cells as well as their progeny cells. Then, using a technique of non-radioactive in situ hybridization, chondrocytes expressing a specific gene of interest are then stained purple. The selected genes that we chose to study included collagen 10, which is a marker for hypertrophic differentiation, as well as lubricin or protoglycan 4, which is a marker for articular cartilage. We also studied collagen 1, a marker for fibrous cartilage, and collagen 2, a marker for hyaline cartilage. So here are the images showing post-op day 0 in both low and high magnification. The image on the left is gross. The middle one in the middle is Masson staining, and the images on the right are the in situ hybridization with purple, highlighting the genes of interest. And then here are just some magnification of the in situ hybridization. Here are images in similar format showing post-op day 7, also in low and high magnification. And then finally, at the last experimental time point of day 28, we did the same analyses and saw that at this time point, there were no hypertrophic differentiation that occurred in the growth plate cartilage that we had placed at the joint surface based on the lack of collagen 10 expression. Instead, growth plate samples which were transplanted to the articular surface with time became articular-like and expressed the gene lubricin. So this suggests that the microenvironment at the articular surface inhibits hypertrophic differentiation and supports articular cartilage formation. To further explore this new hypothesis, we took rat chondrocytes and pelleted them, cultured them with and without synoviocyte conditioned media, and then tested for either hypertrophic versus articular cartilage differentiation using histology as well as real-time PCR analyses. In short, hypertrophic differentiation was inhibited and expression of the articular surface marker lubricin was upregulated when the chondrocytes were exposed to synovium or synoviocyte conditioned media. Some of these genes were confirmed, again, using microarray and real-time PCR. So taken together, the study supported a mechanism by which synoviocytes, through secretion of either one or many factors, directly act on chondrocytes to either inhibit hypertrophic differentiation and endochondral ossification, or promote articular cartilage formation. That is a schematic in the middle there. So that is quite a whirlwind tour of my past life as a basic scientist. I will admit that even though I completed my degree, the work still feels unfinished. Somehow I feel like I ended up with more questions than I started. And perhaps this is normal in research, particularly basic science research. And my mentor, Dr. Ola Nilsson, would say that is why it's called research. So I also like this quote by Winston Churchill, which is copied inside my published PhD thesis. Success consists of stumbling from failure to failure without loss of enthusiasm. So my research experience did teach me the importance of persistence, that no experiment is perfect, and that it is extremely difficult to definitively prove something or anything really in science. Yet the challenge and the uncertainty can be exciting. And if you have patience and can endure delayed gratification, you may get the excitement of being the first person to observe a specific phenomena and then be able to share it with the world. So overall, my experience studying the chondrocyte influences today my clinical practice, particularly in how much I respect and consider the growth plate and articular cartilage when treating patients. I love that growth plate cartilage allows for normal skeletal development, including height and body frame, whereas articular cartilage allows us to enjoy an active lifestyle and to participate in sports, be mobile. On the other hand, we see numerous pathologic conditions related to growth plate and articular cartilage. Many of these do not have perfect solutions, even with surgery. It will be a holy grail in orthopedics to be able to prevent or fully manage these cartilage-related problems. So the next part of my journey was orthopedic residency and fellowship training. And I will say that besides gaining knowledge and skills through training, one of the things I value most from these experiences were the relationships I formed with my colleagues. We are never alone. We can phone a friend. And since starting practice, I've done it on many occasions, both in clinic and in preparation for surgical cases. I will also not be here today without mentors at every stage of my education and training. These are people who have supported my career, gave me guidance and feedback when I needed it, led by example, such that I would want to emulate, and or imparted wisdom. You have the privilege to do 5 things in your career if you choose: patient care, education, research, team coverage, and administrative leadership. Always live and lead with the wind within. Bert Mandelbaum. So I think these words are applicable to those who are pursuing to become a surgeon-scientist-educator. This entity can be defined as a surgeon who is engaged in patient care, translational bench research, as opposed to just clinical research, as well as educating trainees. And to be clear, it is not a surgeon performing bench research in isolation, because I don't believe those exist anymore, but rather a surgeon incorporated in a multidisciplinary team, investigators, each with unique contributions that they bring. It does require a special personality and strong personal drive, because it is not an easy path. It has many barriers to success. I have written down many quotes and words of wisdom from a lot of my mentors throughout my training that I'll share with you guys for the remainder of the talk. Scientific research is harder than clinical practice. In science, we do not have the luxury of consulting colleagues for solutions as we do in the clinic and operating room. Be a surgeon first. For surgeon scientists, no one cares how prolific we are in research if we cannot operate. So before performing a literature review to see what others have said, I tried to come up with my own list of challenges for an aspiring surgeon-scientist-educator beginning in orthopedic practice. And this is what I came up with. Number 1, first and foremost, I believe having the passion, enthusiasm, and energy to do research while also taking the utmost care of patients is the number 1 challenge We have long hours in the clinic and increasing pressures to increase volume and pay attention to our outcomes. Second, creativity and curiosity to generate new ideas is hard. It's hard to come up with a new idea. A lot of things are recycled and or modified and to solve unsolved problems in our field. Understanding and being up to date with both scientific and clinical literature. This requires some training if you are doing basic science in your specific specialty. Have supportive mentors and be part of a multidisciplinary team, because in this day and age, we can't just work on an island. We get further if we have people around us. Be active in national societies. Similarly, it's important to stay in touch with people who are doing the same type of research as you are. Protected time for research, administrative work, as clinic and admin can bleed into time that you have remaining to do research, taking away energy and your enthusiasm, essentially. Grant applications and financial support once your research takes off the ground. And finally, work-life integration, or just life in general, as well as social support for your own career satisfaction and ultimately longevity. So pertaining to work-life integration, it's okay to drop a ball when juggling multiple responsibilities, but it is important to know which ones are bounceable and which ones will break. And Van Heest. So reviewing the literature, the aim of this study was to examine the challenges confronting surgeons performing basic science research. Specifically, this was a large survey of over 1,000 academic surgeons who are members of the Association of Academic Surgery, as well as the Society of University Surgeons. This study found a 27% decline in NIH funding for research in surgical departments from the study period from 2007 to 2014. And then the surgeons who participated and responded to the surveys most often cited that the barriers to success in academic research or pressure to be clinically productive, excessive administrative responsibilities, difficulty obtaining funding, and the desire for better work-life balance. And then, interestingly and somewhat concerning, is that 68% of respondents, including 75% of department chairs and directors, did not believe that surgeons can be successful basic scientists, cannot do two things at the same time. This follow-up study sought to provide a strategy to increase the chances of academic success for surgeons by providing anecdotal evidence. Identified factors such as supportive environment, access to collaborators, supportive staff, committed mentors, financial support, and social support, including family and friends, and being able to set realistic expectations amidst your personal life. Many of these essential components of academic success for surgeons essentially are tied to having the right people around you, it would appear. So I cannot do the things I do without having the best people around me. This includes partners, fellows, physical therapists, radiologists, operating room, and clinic staff. Yellow trash. So the last part of being a surgeon-scientist-educator is educating. This is a review that highlights various strategies that educators may find useful to improve their teaching skills in orthopedic surgery specifically. It talks about active teaching styles, setting clear expectations for trainees, breaking procedural skills into smaller steps. Above all, however, I believe that simply valuing the importance of imparting to the next generation both knowledge and skill is what makes a great educator. Understanding that we have all been trainees at one point in our careers, remembering what it felt like to make mistakes, knowing that failures do occur and that is a normal part of learning. And that the most important point is recognition of the error and the response that is characterized by open communication and timely feedback. When you encounter a complication, the first place you should look is in the mirror. Dan Zuccato. All right, so coming full circle, I will end on talking about one of my current research interests, clinical interests related to pediatric orthopedics and sports medicine. This is a clinical project that relates to my basic science background, continuing the common theme of cartilage research. So this is a histological slide of an 8-year-old human cadaveric donor with osteochondritis dissecans. It shows early cleft formation in the epiphyseal cartilage along a necrotic cleft shown by the arrows. So if you imagine that if the subarticular physis fails to ossify, there will be an island of necrotic epiphyseal cartilage within the subchondral bone, depicted there by the asterisk, which provides poor biomechanical support for the overlying articular cartilage, whereby subsequent repetitive trauma or stress can create an OCD lesion, stable and or unstable. Classic radiographic findings is a variably ossified progeny fragment separated from the parent bone by a crescent-shaped radiolucent line. There are also different MRI findings that can relate to changes in the overlying cartilage, parent-progeny interface, progeny ossification, which is what this project is interested in, as well as parent subchondral bone reaction. So a difficult clinical question remains, when should we operate on OCD lesions? And the purpose of this study, which is the study is currently unpublished, is to examine knee OCD that present with no ossification within the progeny fragment, and to identify patient factors and lesion characteristics that correlate with the eventual formation of bone within the progeny. It was hypothesized that knee OCD that form bone within the progeny would have improved healing rates in clinical outcomes compared to those that do not form bone within the progeny. And the significance of this is that knowing specific patient factors or lesion characteristics that indicate bone formation within the progeny may help guide decision whether to non-op or to operate for patients. So this was a single study, single center study, patients that were prospectively enrolled into a knee OCD cohort. Inclusion criteria: knee OCD, no bone within the progeny, and greater than 1 year of follow-up. So the two groups were no bone to no bone in the progeny, and no bone to eventually forming bone within the progeny fragment. Data collection for the demographics, X-ray, MRI, healing status, patient-reported outcomes. So here are examples showing OCD lesions on the top that started without any bone, underwent antegrade drilling, and then ended up with no bone in the progeny. The example on the bottom shows an OCD lesion without any bone, underwent transarticular drilling, and eventually healed with bone bridging the progeny and the parent fragments. So the results were as follows. There were 29 patients in the first group, no bone to no bone, and 47 patients in the second group, no bone to bone formation. And the results, the only significant difference in demographics in this study was that the patellar OCD lesions did not form bone in the progeny. The only significant MRI finding was the interface signal between the progeny and the parent had significantly less signal compared to the fluid in the joint space. So this perhaps indicates fibrocartilaginous healing, then that leads to bone formation. There were no differences in patient-reported outcomes between the two groups that were significant. So at the moment, conclusion of this study are the following: that MRI finding of the interface that had less signal than that of the fluid positively correlates with bone formation in the progeny. However, the healing rates and the patient-reported outcomes do not significantly differ between progeny that subsequently form bone and those that persist without bone. As with many studies, larger numbers will be needed to detect statistical significance and to determine clinical value of knowing whether bone is present in the lesion. So I left some time for questions and discussion. It's just appreciate your attention and also the opportunity to share about myself and how I came to join you at UCSF. So, thank you.
Thank you, Dr. Chow. That was a great journey, and welcome to the group here. I think we have some time for questions and discussion. So Dr. Sabra will sing that.
Michael, thank you so much. That was really very insightful. And, you covered so many broad themes. And firstly, I want to acknowledge and say we've enjoyed having you here. You're so thoughtful, calm, humble. So my first is really in support of your observation and to see if you have any sort of fixes for that. In my previous job, we had two residents who really came in, both were PhDs, wanted an academic career, and they both ended up in private practice without a fellowship. So, and we heard earlier today about burnout. And I just want to know, if there's a part of that happens in a clinician scientist's life, especially in surgical specialties, and what, if anything, is a fix? My other question is totally unrelated to the first, which is, recently I've been exposed to, and we've been doing some studies on DTI, diffusion tensor imaging, for growth plate issues, which is really like a functional MRI of the growth plate. And, that would be a nice translatable thing from basic science to clinical practice. And how do you see if, you've got the exposure on how DTI can sort of evolve into a clinically relevant imaging?
Thanks, Sanjay, for the two questions. So to answer your first question, I think it largely depends on the individual and their real passion or interest in doing research in the first place. I think in this day and age, when we applied for residency and fellowships, research is required. So some people do research, as like a stepping stone. But even those with PhDs, and or have gone through like a MSTP program, like you said, may not continue their research in the future and go into private practice. I don't know of a fix for that, aside from maybe putting the right people around them when they go through training, introduce them to people who have done similar work that they have in the past, perhaps like just keep their interests in their passion for research kindled throughout their training. Burnout is real, and I know people who drop out of MD PhD programs and or just change direction 180 after having like 4 years of experience, which can be seen as somewhat wasted. But yeah, I think putting the people around them and just encouraging them to continue their work is probably the one thing that we can do. And then your second question, I'm actually not too familiar with that form of imaging. But I think that, at the University of Minnesota, they had an MRI with the STAR protocol that can also look at cartilage in detail. It's not yet used in clinical practice, but only for research. But the imaging that you suggest sounds like it's further along. So I'll be excited to find out more about it.
Other comments or questions?
Thank you so much. Welcome to the department. First, a comment. Should not be discouraged by this talk because there are people who have accomplished what you're describing. The traditional triple threat is not easy, but it's doable. And I do believe that not all orthopedic surgeons can do it, but there is always a subset that will be successful with basic science and clinical practice. You do need all the factors that you mentioned. Departmental support is really critical. Protected time is essential, especially early on. But it is doable. I think we have faculty members who've done that. We have current residents who I'm 100% sure will be successful in that role. And UCSF may be an outlier in facilitating that path. But I think now you are in an environment where that is definitely possible because all those factors you mentioned are present here, along with the institutional commitment to having people be successful in that role. That's essential. So don't want any of the residents to believe that it's just not doable anymore. It is. It's hard, but it's definitely doable in the right environment. Tons of questions, so I'm not going to rattle off 200 of them. But the Dr. Kuo has done some research where you take articular cartilage, implant it, use it as graft, and it ossifies. So it's the kind of the other side of the coin. You take growth plate cartilage, flip it, expose it to synovial fluid, and it becomes articular-like. You can take articular cartilage, flip it into an environment that's primarily bone healing. It will form bone. So the question I have for you really is, does that make sense in your paradigm, first of all? And number two, is it direct kind of endocrine signaling? Is it spatial organization? Is it mechanical load? What do you think would be the main factors, and what do you if there are diffusible factors like you would assume from synovial fluid? Were you able to identify any of those key factors? Because that would be the natural, holy grail if you could find the things in synovial fluid that actually stimulate articular cartilage.
Thanks for the comment and then the question. So there was a follow-up to that FLIP study where we, instead of taking growth plate cartilage and put it on the articular surface, we just took articular cartilage, immature articular cartilage, and just flipped it upside down. And in that scenario, as you, I think, are suggesting, the deep zone of the articular cartilage does not turn into superficial zone chondrocytes, but rather does ossify. That's unpublished data right now. So I think there is some extent of kind of a predestiny for the chondrocytes. Like if they are already terminally differentiated, for example, it's almost impossible for them to turn around. I think in addition to growth factors, say within the synovial fluid secreted by the synovium, biomechanical shear stress, I think it's important. The lack of axial loading, I think it's important. And also endocrine factors that are kind of hard to test within the lab. So I think it's multifactorial. I think that potentially we can find maybe one or a family of factors that can encourage the stem cells or stem-like cells to differentiate into articular chondrocytes. And with the pellet culture experiment, that was essentially what we wanted to do, was to isolate the conditioned media and then purify as much as we could and then use that to see if we can differentiate the chondrocyte pellets into articular cartilage-like tissue. So that's still kind of in the works right now, but I think it's potentially something that we could study and find in the future.
Any other comments, questions? Dr. Ma?
Mike, welcome. Welcome to the department. Actually excited about, seeing some of the work you've done before. We chatted before about that also, but it's great to kind of see in the presentation there. I will kind of follow with Dr. Kim's comment. This is what this department values. We value, scientists, we value educators, we value clinicians in this department. And we have a lot of successes over the years, some of them in this room already. So I would not let the passion die off in whatever way that we could kind of help to continue with those activities. Please tell us, because this is important for us to continue to be like innovators and also leaders in this field. On another note, let's switch, gears a bit over there. We do see these OCD quite a bit. Obviously, you're looking at the mechanism about, why some of them heal versus the other, and what the prognosis are. So my question was, why do they occur at the first place, and why at those locations? Because a common, scenario is always lateral aspect of neurofemoral condyle, central posterior lateral femoral condyle. Why do they occur in those areas? And do you have any insight about, mechanism of, injury? And clearly something's happening and we're not really solving the problem right now.
Yeah, thanks very much for the comment and the question. I think it's idiopathic still. We don't know exactly, but we have lots of different theories. I think the leading theory right now of how OCD occurs is a combination of repetitive stress and also poor blood supply. In my mind, so you mentioned the central aspect of the medial femoral condyle as being one of the classic sites. In my mind, the femoral condyle is very square shaped, and so there are a lot of areas where there can be watershed areas. At the same time, also get hit repetitively, say by the tibial spine. So I think that's a very likely explanation. And also when we look at OCDs in the elbow, there are different locations where OCD occur when you compare gymnasts who are axially loading their elbow versus overhead throwers who put their arm in that slot position. The OCD lesion occurs in different spots. Furthermore, if you look at catchers who are always in this deep knee squat position, their OCD lesions occur more posteriorly in the femoral condyle. So I think it's hard to prove anything exactly, but there are a lot of good association studies that would suggest that mechanical and vascular are possible causes. And then you have genetics, and then you have endochondral ossification of the subarticular physis, and metabolic things, vitamin D, et cetera, that could play a role as well. So I think it's the jury's still out, but I think there are some good guesses.
All right. Any other comments, thoughts? So I think we'll look to wrap up. Dr. Chow, thank you so much. And again, welcome, and look forward to seeing your ongoing work.